morning to everyone present today for the first lecture of the fourth day of the workshop. I really appreciate you all for attending today. For the hands-on session, we have shared the links in the description box. I request everybody to please have a look on that. Now, moving forward, may I request Dr. Yasha Hasija to please introduce our first expert speaker of the day, Dr. Tannistha Nandi. Ma'am, please.
A very good morning, everyone. I'm extremely glad to introduce you all to the expert of the day, Dr. Tannistha Nandi. Experienced data scientist with a demonstrated history of working in the research industry, Dr. Nandi is skilled in HPC cluster, databases, machine learning, bioinformatics, genomics, and life sciences. Dr. Tannistha Nandi is currently working at University of Calgary, Canada as a data scientist. Prior to this, she's worked for 8 years at Genome Institute of Singapore as adjunct staff scientist, and research associate. Dr. Nandi holds a PhD degree in bioinformatics from CSIR IGIB, one of the pioneer institutes in India. She is a prestigious alumnus of Indian Institute of Technology, Roorkee, for her master's degree, in which she was the first rank holder. Down her career lane, she holds 23 publications and 1 patent to her name. Dr. Tannistha has also received Long Service Award of 5 years from Genome Institute of Singapore. I now invite Dr. Nandi to take over her session on data visualization with R. Dr. Nandi, please.
Thank you, Yasha, for this kind introduction. I'll present my slides first. And sorry, it just took me a minute to get started. Good morning, everyone, and it's a pleasure to speak with all of you today. I'm really excited to meet all of you and be a part of the DTU workshop. And what I'm going to do over the next 2 hours or so, I would like to give you an idea of some of the things that you are able to do with R, specifically in terms of visualizing data. And throughout the session, I have included several references that will explain some of the additional details, give you a background and work that has been done in this field by a wide number of research groups. And what I have done when I have structured this talk, I have interwoven throughout, it's kind of a mixture of topics that are conceptual and introductory with the ones that go into little more details about how processes would be carried out. Most of it I'll talk through. Some of it, if it doesn't resonate right away, don't worry. You will have these references with you to go back and check again. So there's a fair amount of material, and hopefully we'll get through all of it today. Okay, a few housekeeping things before we get started. I have shared the data set that I'm going to use for for today. And so this is an actual data based from National Health and Nutrition Examination Survey. So I'll use this case study and I'll run through a lot of steps and commands. Not every single one of them, but many of them in terms of getting data organized and using visualizations to get insight into what's happening in the data. And the other 2 things that we will be needing for the hands-on session are R and R Studio. So here you have a choice. You could, if you would prefer to watch me doing the steps and launching the commands, which is totally fine. Or if you like, you may also try them as we go along the course. Okay. So talking of visualization, any programming language or application is first and foremost a tool. So I have this picture up on the slide showing a Swiss knife, and you can see there are different attachments. It could be a scissor, knife, filer, whatever you can think of. And you would like to bring out the right tool or the attachment for the job at hand. Okay. So in a similar fashion, you have a range of tools available for working with data, and one of which is R. But there are other tools too. And I want to show you where R should be in your data science toolbox. Okay. So when we talk of data, I'm sure all of you have worked with spreadsheet just because they are everywhere and everybody uses them. And some of the common choices you are familiar with will be Microsoft Excel, Google Sheets, and there may be other possible choices as well. And the reason these spreadsheets are great tools because they let you organize your data the way you want. You could do some tasks like you could sort, you could, let's say, filter your data, you can have a quick summary, you could have counts, and you can quickly have a graph and have some sort of a visualization. Okay. And truthfully, the spreadsheets are probably sufficient for some of the data tasks that doesn't involve very high level of creating models and statistics. But when we are talking of scientific research, especially when we are talking of bioinformatics, which is an expanding field, we have data from like a huge omics data set, right? Genomics, transcriptomics, metabolomics, and so on. It's getting into that big data industry. So that's why it's a time to move beyond these summaries and basic graphs, and we need to get a more sophisticated tool out of our toolbox. And when I say this, I'm sure some of you are thinking, like, which tool is the best? Is it Python or is it R? I think we had this discussion yesterday, and I'm very glad Dr. Gunjan has explained this in a very nice fashion. So I would just reiterate that this isn't a very helpful debate. And to summarize, what I would like to say is that both of these, R and Python, have very important strengths. Python is especially strong in machine learning applications. And think of R, it's very strong in data analysis, which is very different from machine learning. And that's why it is a very popular tool in scientific research. So with that, let's get started. I'm sure some of you might have R already installed on your devices, but just for those who don't, I'm going to show you how to install R Studio and R programming language onto your computer. And it's going to be real quick. And it doesn't matter what type of operating system or device that you're using, whether you have a Mac or a Windows PC, the steps are going to be very similar. So if you just follow along, we'll get sorted in no time. So the first thing, what you need is, as always, a browser. And what you have to do is you have to point your browser to the website, which is on the top, cran.r-project.org. And when you get to this page, you will see a link to download R. Okay, and click on that. Depending on your operating system, choose one of the three options presented if you have Windows or Mac or Linux. So once you click on the download, it will start downloading the application for you. But whichever approach you use and whatever operating system you are going to use, it's easy to download and install like any other software which you have. So once you install this, you will have R on your machine. So for today's presentation, I have R version 3.6.3, but you can choose any of the versions that you feel appropriate. And the other thing, what we need is R Studio, which is a separate application. It is an environment which is developed specially for working with R. It is a very convenient way for the data science type of projects. So now to get R Studio, you have to go to the link, which is at the bottom of the slide here, which is rstudio.com. And then come right to products. And you are going to choose R Studio, which will land you to the page here. So now there are two versions. You want the desktop and not the server version. Okay. So again, this is open source, which is free. And once you get there, you click on download R Studio. Once you do that, then you get to come down and make that choice one more time. Okay. So we still want R Studio desktop. And once you do that, it's going to automatically detect your operating system and hopefully offer a button to download. So once you see that, just click on it. And again, it's going to be a quick and easy installation like any other application you put on your computer. So now we have two things with RStudio that you have hopefully downloaded right now, and the R application that you downloaded a minute ago. You will be ready to start working on the data. Okay. So once you have got RStudio open, you will actually see a collection of window panes. Now, the nice thing is that it's actually a single window and it keeps things organized. Sorry. It breaks it up into logical components. So up here on the top left is where you will have your scripts or programs that you write, and then you can save them. You can have more than one open at a time. So basically, you can open multiple scripts. And just below that, here I have a console. This is where your text or numerical output will go. Okay. So any command that you execute, the output will be shown in on this console. And then off here to the right is the environment. This is where you will see any data set that you have loaded into the memory or the variables that you have created as you start using R. So all of them you should be able to see here. And you also have a tab for history where your commands that you have executed previously. And if you are connected to a server or maybe to GitHub, you have those connections here under the connection tab. Okay. And then down below is the last pane. So here, for example, I'm having the files that I'm going to use for this R project today. And you have quite a few tabs here. You will also see a list of packages that you have. Then you will have a viewer, which means like any plot that you that you do with your data will also show up here. You have a help function here, and what more things you have? Yeah, so actually there's a lot to do here. You can explore at leisure, and it's fun, but I will not get into that today. But this is the overall idea that at high level you need to know to get started. Okay, oh, before I get there. Okay, so when we talk of R, it's basically a collection of packages. And what is a package? They are all R functions. So the way it works is, you have different packages which you load at as and when you need as you go, and it increases the power of R by improving the existing base functionalities, base R functionalities, or it could add some new functionality. And we will see how it goes. So one of the things about our packages is that it contain other packages. So it's kind of a complete self-referential thing. You don't have to bother about the dependencies as it goes. It's self-inclusive. Okay. So I'm going to show you the use of this. We are going to use a lot of them, but one of the important ones is this tidyverse. And the reason I'm having up this on a slide is because, I'm going to show you how it dramatically changes the way you work with R and improves the functionality. So in fact, if you want to go to this, to the details of this package, you can visit the website, which I have down here, which is Comprehensive R Archive Network. And then click on to the packages. And it's a searchable thing. It will bring up a huge list of all the packages that are available. Search for tidyverse. Okay, and if you go to tidyverse, I don't have it up in front of me, but if you go there, you would see that there is a website called tidyverse.org. And if you click in, actually, it will explain a little more what's going on. And it will show you the packages which are there under this tidyverse package. And if you take a look under packages, you will see that it includes ggplot, which we are going to use today. And it adds up a lot of functionality. Some of you might be familiar with packages like dplyr, which is very useful for data wrangling. So everything is packaged under one umbrella, which is tidyverse. So hence it's pretty useful. And we are going to use this today. So with that, what I'm going to do is I'm going to now switch on to the hands-on demonstration, and I'm going to bring up my RStudio, and you guys can bring up yours, or you can just see when I execute and how do I bring up the graphs. Okay, so I'm going to end this presentation here and switch to the demonstration mode. Do you guys see my RStudios?
Yes, ma'am, it is visible.
Okay, that's great. So this is the RStudio environment that we have just seen a screenshot in the presentation. So what I'm going to do is I'm going to write the scripts here. Don't worry about the individual commands. I'm going to pass you the script end of this workshop, end of this session. So you will have it. But just try to understand the way the workflows work and how do we decide what we need as we go along. Okay. So the first part, I'm going to show you what are the different ways that you can install and load packages. As many of you are just getting started, you would be preferring perhaps the graphical user interface, and that's exactly what RStudio provides you. And if you go here to packages, what you see is that it brings you all the packages that you have. And if you want something which you want to install, easiest way would be click here, install, and it is actually bringing you to the same repository, the website which we have just visited, CRAN repository, and you can actually name the package you want to install. And you could actually install multiple packages. Let's see, Pacman is a package manager. And once you have all the packages that you want to install, you could just check this box. It is often better to have the dependencies installed.
Excuse me, ma'am. Sorry
Yeah.
to interrupt you, but ma'am, participants wanted to know that if you can increase the font size of the this thing of the RStudio, if it can be done.
It's maximized. Let's see. Let's see. Is it any better?
I guess so.
I'm not sure if it is. More time. How about?
Yes,
Okay, great. Yeah, it's all good. So basically, you just push install here, and then it should automatically install it for you. But I'm not going to do this way. I'm going to show you the command line way of doing it, just because that gives you a little more flexibility, and at some point you should pick it up. So I have them here. So what I'm doing is I am installing and loading a package which is called Pacman, which is a package manager. Okay. So what this command basically checks if I have Pacman already installed, or if it is not there, then it's going to install it. Okay. So that's a condition which I'm able to do, that flexibility with my command line, which I am not able to get if I just go to the install with a graphical user interface. Okay. And the way to run this is just select it, and you have a run button here. So it will execute it. Just because I have it installed, so it didn't reinstall it for me. But if it wasn't there, then it would have installed the package for me. Okay. So what I'm going to do now, because this is a package manager, using this, I'm going to show you how to install, how to load other packages. So I'm going to load a bunch of them for demo purpose. So I'm trying to include as many commands as I could. So what I'm doing is I'm saying Pacman to load these 4 R packages for me. The reason I load Pacman is because later I will show you, now I am loading the packages. Later I'll show you how do I unload these packages. I can unload all of them at the same time, or I could just unload the one which I don't want to use at that point. Okay. Then I have the psych package, which is for statistical procedure. You may have something else, whichever is your preference. And then this is RIO for input output, basically for importing data. And then tidyverse. So today we are going to look at few functionalities of tidyverse, but it is not limited to only those. It has many other functionalities. So basically, I'll be showing you piping and visualization, but you can explore. There are many other functionalities to tidyverse. So now I'm going to execute this command. Just to mention here, anything that has this pound sign is a comment, and that's basically for our reference to, it's a kind of annotation that helps us to understand what's happening in the script. Okay, so it has loaded all these 4 packages for me. The other way to check it, if you scroll here in the packages, you will see that there's another way. If I want to load, I could just check it here. Then also it will load the package. I can detach the package just by unchecking the box. And in case if you don't want to use the package manager and you want to load the packages manually, that's possible. You just call it through this function called library and name the package. This is another package, which is basically the base package of R. So it brings up, so these are a few ways that you could install and load packages. Now, another thing is more of a best practice, or is to basically have your working directory set. So meaning that instead of browsing the files and giving different paths to your input data, you could have everything under, let's say, one folder, which I have here. And I could set my working directory and pointing it to this folder. So you could set it either by going here and you could choose the directory, browse the path. This is again a graphical user interface way of doing things. But if you want to do it the command line way, so all you have to do is you have to use the command set wd, and I'm giving it the path to this directory to set the working directory. So it will ensure that, okay, now I am in the right directory and I can directly read in any file which I see here. So now next what I'm going to do is I'm going to import this data set. Probably you all have the link to download this data set. This data is from a survey that measured the iron status markers in children aged 3 to 5 years old. So I'm going to import this data. So that's fairly simple. All that you have to do is you have to name a variable into which you are reading the data. The function that we are using here, it's a CSV file, comma separated file. So we are using the function read underscore CSV to read in this data set, okay? So, and I have to execute it. And you see once it has imported the data set, it appeared here under environment. So this is something which it is there in the memory. And so all the data and the variables, you'll see it will get populated here. And if you want to take a quick peek into what's happening in our data set, right? We just usually do this. Head data is going to give you like the different fields that you have and the rows of your data set. And one very important thing which happens in R pretty often, because we are working with um, big data sets, sometimes depending on the RAM which is available to your machine, the entire data set doesn't get loaded. Okay, so you have to ensure that you have loaded the data set completely before you start your analysis. There are a couple of ways to do it. Firstly, you can look for the dimensions here. So you have 10 variables, which you already probably know that you are expecting 10 variables in your data, and you can make sure that everything is loaded into the R memory, and then how many observations that you have. So in this case, we have 295 observations. Um, the other way, if you want to check, is you could go for this command, dim, which stands for dimension, and it's going to give you the rows and columns. Okay. Couple of other things which I'm going to show you, just so that you have some, how do you explore the data when once you are given or you are getting started with a new data set. A command that I like, and I use it quite a lot, is table. And I'm going to show you how it works. So what I'm asking table that, okay, can you tell me, let's say, um, I want to look at the different race or ethnicity that I have, and what is the distribution here. So this will give me a nice summary that this shows that there are 5 different ethnic groups, um, and then their individual numbers. Okay. Similarly, you could do it for any other variable that you like. Maybe we always look at the gender distribution. Maybe that's another thing we could do here. So it tells you the distribution of number of males and number of females. Okay. Then I'm going to show you how, so the way I'm going to do is I'm first going to show you some of the things that we can do with base R functionality. And what are the different types of visualizations that you can do, and how that can help you with your exploratory analysis. And then we will move on to a more advanced way of creating different visualizations. So let's say, just to start with that, let's say I want to know the sum of all the values that appear in this field in the called subject. So I'm using this function sum, and I'm passing this argument that field subject from my data set called data. Okay. So it gives me the sum. Okay. Now, what I'm going to show you next is how you can pipe commands in R. I'm just going to write it here, but this is not something which I'm going to execute. So this funny little series of characters, the percent signs with greater than symbol in the middle, this is known as the piping character. And this is included as a part of the package tidyverse. Now, there's a reason that I am talking about this, because I'm going to show you how it changes the way you write your code. I'll give you an example of a command and how you would write it in base R. So when I say base R, it basically uses nested commands, which means you start in the middle. The way you read the command is you start in the middle and then you go out. Okay. So for instance, let me bring up a command that I am going to show you. Hang on a sec. Do you guys still see my screen?
No, ma'am.
No. Okay.
See,
I think some. Just give me a second. Just went away.
yes, ma'am. Now it's back.
It's back. Okay, that's good. Okay, so yeah. Okay, let me just put this as a command, as a note here. This is the piping character. Okay. So we are, okay. So the way, so this is a base R command, and it is a nested command, so which I was saying. So the way you have to read this is, we'll start from the middle. That is the data, which you see here. And then this data, as such, when we have read it, this is a data frame. That's our object. But the calculation what we are doing right now, we want to read it as a matrix. So I'm converting it into a matrix here with this command as matrix, and then passing the argument data into that. And then what I'm trying to do here is, okay, how do I say this? I basically want to compute the percentages of the data that are present in the table. Okay, so I'm going to use this command margin.table, and then I'm saying whichever margin I want to use. If you want to know what does this command do, you could just write a help here, margin.table. It should bring up the help. It's basically compute the sum of the table series, whatever index I pass it. Then prop.table is another command to compute the proportion. So probably it will, so it's going to compute the proportion of that variable. And then I want, it will be a too long decimal character. I want to round it. And then if you again come here, I want to round it up to 3 decimal places. And then I want to convert it into percentage. So I am multiplying it by 100. Okay, so this is really a nightmare to understand what's going on in this command line. But this is how base R functions. And I'm going to execute this, and you will see what you get. Okay. Now, if I show you the same thing, how you would do this in tidyverse, you will really appreciate how simple it is to understand what's going on. Okay, so now this is a tidyverse version. So the first thing is I am reading the data here as a matrix, then piping this data into the margin command, piping the output of margin to the proportion. Then I'm asking it to round it up to 3 decimal place, and then converting into percentage by multiplying it with 100. And I'm going to get the exact same output. And so that's why I'm saying that if you use these R packages, it improves the functionality. And it's just that you need to know like which package could improve your functionality over the base R. And there are different packages. Maybe one is better than the other, depend on your comfort level. But tidyverse is one of the packages which is very popular, and it's pretty useful. Okay. Now, I'll show you another example with this data set. So this time I'm going to show you the tidyverse way first, and then I'm going to, what's happening here? Yeah, I'm going to bring up the example for the tidyverse way so that you understand what's going on. And then I'm going to show you how we will do it in base R, okay? So let's talk of another example here. So what I'm going to do here is I'm reading this data that we have here. I want to filter it. I want to only look at the race or ethnicity. And we want to look like race and ethnicity per the ratio of male and female. And then the variables we want to work with is the ethnicity and also like the two markers that we want to look at. And then we want to look at the distribution and group by the ethnicity. And finally, we want to summarize it. And the way we want to summarize it is we want to use this function, use a mean function, okay? And so this gives me the summary of what I have requested. Now, if the same thing, I have to do it in using base R functionality without the tidyverse package, it's going to be something like this. Okay, let me put this down. So I have to make use of other things. Basically, I am using a condition which here. So I'm saying, you know, which date, which race or ethnicity is in 1 or 2. Sorry, it's not the gender here because we have 5 different ethnicities and I only want to select for the 2 ethnicities which are coded as 1 and 2. And then I want to look for these 2 markers in the data. And then I have to apply this lambda function to compute the mean. But at the end, I'm going to get the same result. Okay. I just wanted here to, the takeaway point here is that if you use the right package, it could save a lot of time writing these codes. It's really easy. The way you think, you want to go and filter or summarize your data, you just pipe it the way you're thinking in your head. We want to filter first, then we want to select, and then want to group by, and then whatever function that you want to that you want to use to summarize mean, median, or frequency, anything that you're looking at. Okay. So with this, then I'm going to move on to the next part, which is where I'm going to show you some basic visualizations. So before I get started, I'm going to load one more library just because it will make the plots pretty, which is called color tools. It has a lot of I'm going to use it to pull up some colors. Okay. So when we are talking of visualization, the purpose is actually to look at your data and see what is happening. So the easiest chart for the most fundamental kind of data is bar chart. I'm sure all of you know what a bar chart is. Simply see how common something is into the data. So what I'm going to do next is I'm going to plot it, and using a generic, again, base R bar plot command. So I'm simply going to take the data, select the variable, and plot them. Okay, so let's see what do we get. Again, these base R functionalities, it's adequate for any exploratory purpose. So if I have to do a bar plot using the data that we have just read in, okay. So what I'm doing here is first I'm using this command table to generate the numbers, the distribution that I need. So if you want to see what is there in the number, we could just run this part of the command to see. So it's giving us the distribution and we want to plot this as a bar plot. So the simplest way to plot would be, I'll just do this first to show you. If I don't do anything, I just pass this data to the command bar plot. It's going to bring up a plot which shows you, at least shows you what you need. Now, if you want to make this plot pretty and you want to annotate, so you can add a title to the plot, you can have a label for your x-axis and y-axis here, you can add a legend, and also you could choose colors for each of these groups. So that's what we are going to do here. And yeah, so you get a much prettier visualization using just base R functionality. Okay. Now, when you have something that you are measuring, that is, you have a quantitative variable, and probably another useful graph you can make is a histogram. So that's what I'm going to show you next. So just remember, whatever we are doing now, these are all base graphics in R. We haven't used any package like ggplot2 or anything, just because they are quick and they are built in and they are for exploration. Okay, and they are usually exactly what you need to look at the data. So I'm going to give you some, for example, okay, let's say we want to do a histogram. So the command for this is hist. And if I want to know how do I use this command, you just have to look for help and it will bring up the help. But I'm going to first show you just a one-liner. All that you need is hist. And again, referring to the data set that we are going to use, and we can actually choose any variable that we want to. Okay, I'm just choosing this one, hemoglobin, and let's see what do we get. Right, so it gives you a histogram, and then again, if you want to make it pretty, you could add more commands. There are several options. For example, I want it to be steel blue. I want to fill this color here. And then I want to have a title. I don't want this default title, which just says histogram of basically the variable name from the data set. And also I want to replace the x-axis label here. So I'm passing those arguments to the function hist. And it brings up a nicer histogram for me. Another useful visualizations which we often use is box plot, because something that you are counting or measuring is a box plot. And I mean, because they are very helpful for both simplifying the data that you are looking at, as well as for the presence of any outliers, which we often want to look into our data sets. And we have to deal with the outliers so that it is not skewing up the trend, right? So so box plots are that way very useful. So let's do a box plot. And I'm going to again use this base graphics first. And so what I'm going to do, the same thing, take the data and I'm going to say, give me a box plot. And when we do that, we kind of get a basic box plot. So what we could say is box plot. And then let's say we want to look at the data here, and I am going to choose, let's say, the same variable, okay? So it gives me the box plot. Now, if I want to look at the hemoglobin distribution, but I want to split it, I want to look at it, I want to split it by the gender. So, not months, I have to do it by gender. Okay. And I have a little tilde, which basically says factor it by gender. So that's the little trick here. And if I run it, it has shown the distribution of hemoglobin and by the gender, okay? And again, we could do many other things, make it pretty. For example, we could, let's say we want to look at not the gender, but different ethnicities. It's the same thing, hemoglobin, and I have this little tilde here, but now I am saying look at the race or the ethnicity, and I'm passing, instead of using data here, I'm passing it as an argument, okay? But you could use either ways, both are fine. And then I'm saying label my x-axis as ethnicity, y-axis as the hemoglobin measurement, instead of just the variable name, add a nice title to my plot. And this is basically which controls the font size. Okay. And you can actually see how this can, if you change this parameter, it's going to reduce the font size. Okay. So you could just play around, standardize all your figures whichever way you want. Okay. Then let me show you the other type. Okay, so far what we have seen, they are all basically looking at univariate analysis, looking at one variable at a time. And they're critical, they're informative, and they can be interesting, right? But really where things are more exciting when you look at association between more than one variable. And one of the most basic ways to do that is by looking at scatter plots. And that's what I'm going to show you next. Okay. Because they will, it's a way of showing relationship between two quantitative variables. So let's say I'm going to plot, this is the simplest one. You just have to use a command like plot, choose the two variables. Let's say I'm looking at the iron and then the total iron binding capacity, which is, I'm not sure if it will bring up some sort of a relationship, but let's look at it. So basically, I am plotting my X and Y here. Okay. Well, it brings up for each subject, I have it has given me an XY scatter plot. Okay. Now I can go and ask several other questions. For example, I can subset for a particular gender, and I could again play around with some coloring and some of the symbols that we have used here. I'm going to show you in a second. So for example, here I have added another command, subset gender equal to 1, meaning just subset the males, and then do the same plot, color it in steel blue. And this argument here is to basically decide the shape of the dot that we want on the plot. Okay, so I could go ahead, if I don't like this, I'll show you how, if I change it, it's going to change. And by default, it's an open circle. If you want something else, So there's a whole list of these which are available very easily. If you just Google, you will get all the options that you have to visualize your data in different formats, right? This is the simplest scatter plot. And one other thing would be heat maps. Okay. So heat maps are great tools for visualizing the cluster analysis. For example, a phylogenetic analysis that we do, or any sort of clustering that we are doing. When you have a number of different objects or different variables, and you want to see how they group with one another, a cluster analysis is a great way to do this. It takes the variables in your data set, look for similarities of each case based on those variables. And then if you want to visualize this thing, then heat map is a great tool. So for heat map, we can't pass in the whole data set as such, because we want to look at we want to select the variables that we want to look at. So I'm going to create a subset here. Let me just close this. Okay, so now we have only one data and one variable which is under value here. So what I'm going to do here, I'm going to create a data frame, and I am saying that I need only with these two variables from the data set which we have stored in the variable called data. So just select these two variables, create a new data frame, store it into a variable called data frame. Okay. So now I have only two variables here because that's what I have selected for my heat map analysis. Okay. Another thing which I want to show you here, it's not related to heat map, it could be to any analysis. I want to check the class of these variable, because we just discussed about that we used this function called as matrix a while ago. So if I check this is a data frame because I have combined it using a command called data.frame, which gives me a class called data frame. Now, if I want to convert it into a matrix, as we have used previously, I'll use a command as dot matrix, and I will pass this data frame, which will change the class of this variable into a matrix. So I'm going to show you how. Okay, so let's just do the first command first. So what I've done is I have created another variable data matrix, and I have passed this data frame and asked it to convert this data frame into a class called matrix. And now if I run the command again, oh, I have to run this. Okay, and it has, oh, I have to ask for the class for this. To say class data matrix. Okay. Now this should return me a matrix. So why did I convert it into a matrix? Because the input for the function heat map is a matrix. And the way to check it again will be if you go and look for help, you could get all those details, that what sort of input that you need, what arguments that you need to pass into the function. So now the simplest again, I will pass this matrix to the function heat map, and it should be able to bring up a heat map for me. Not the best, but you could go ahead and customize it further. You could have gradients here, and whichever, as creative as you can get, and you can choose different color schemes and stuff like that. Okay. for the heat map. And then I'm going to show you how we do a correlation plot. So for doing a correlation plot, I'm going to pull up another library, which is called corplot. And this is basically another R package. Okay. Now, what I'm going to do is I want to look at the correlation between all of these variables. So it's going to be a 10 by 10 matrix for me. So first of all, I have to calculate the correlation and then visualize it using a plot. So the first thing is I'm going to calculate a correlation matrix. So I am passing the data to a function called core, which stands for correlation. So it will give me a correlation matrix, which appears here again. And then I'm going to use another function, which is called corplot, to plot this matrix. Okay. So here's the correlation matrix. It's a 10 by 10 matrix. Of course, diagonal is 1 because it's perfectly correlated. And then you have this heat map here, which can tell you the level of correlation. Looks like these two variables are correlated, more correlated than anything else. There is a negative sort of a correlation which appears here. So whatever is the relation, you should be able to just view it just by using this correlation plot function into your data. Okay. So like these are some sort of, these are basically the basic visualizations that you can do with R. And then going forward, I'm going to show you how we are going to create some of the same visualizations using a more advanced package, which is ggplot2. Okay. That's going to be the part 3 for today's session. So, so far we have seen how to do, I would say, some quick and dirty plots with plotting functions like hist and plot, which are built into base R. Okay. Now, the tidyverse has its own paradigm of creating graphics called ggplot. The advantage to using ggplot over the base R function is that ggplot comes with many built-in defaults to make your plots look nice without having to code or too much customization. So as we go through some examples, you will note how ggplot has automatically chosen some color schemes, scales, axis labels for us without us specifying any of these. But of course, we can override these settings. Having some usable defaults built in definitely makes it fast to produce some nice plots. That's all I'm saying. But again, you can go ahead and customize them. So what I'm going to give you here is some of the references, which I really find very useful. I often refer to this cookbook, which has a lot of nice codes which you could customize for your own projects, and you could choose from a variety of visualization from our graph gallery, and then cheat sheet, some nice commands for if you have some idea like how your plot should be, just use them as a reference. And you, even without getting much deep into the programming, you could quickly come up with a nice plot for your data. Okay, now what I'm going to illustrate now, I'm going to use the same data set that we have here, but now I'm going to read it in a different way, okay? I will read the data file, I'll clean it up a bit, and again, we'll take a look at the data. I'll show you what I mean. So we are going to read the data into a different variable now. We'll not call it data, just because we are not going to use the base R functionality. Okay, so now I'm saying it's a NHANES iron measurement data set, and I'm using the function read CSV again because the input data is a CSV file. And then I'm specifying my column types, okay? Like subject is a factor, gender is a factor, like the numbers we cannot really add up. Those are like a subject is a sort of an identity for that patient, or you can think of as your health number or whatever national identity number for that individual. And then gender 1 or 2 is again a factor, right? We can't add them, or we can't do any sort of computation or calculation on those except for using them as a factor. Then age in months, of course, is an integer, and again race and ethnicity is a factor, okay? And now I have defined my column types here. I have piped it and used a function called mutate. This is from the dplyr package, which is also there in tidyverse. And I am recoding the gender. Now, if I go back and show you the previous plots, it's not that informative when if I say gender and then I say 1 or 2, I don't know what is 1 and what is 2, but I would like to have, I would like to recode them. And now I'm using this mutate function from the dplyr package, and I'm saying recode the gender when you see 1 as male and 2 as female. And similarly, I know I have 5 different ethnic groups, and now I want to add the actual labels instead of just having them as 1, 2, 3, 4, 5. What I want to do is I want to recode these numbers into their actual race or ethnicity. So that's what I am doing here. And now if I read in this data set, and so it appears here, okay? And if I'm going to we can definitely use head, but I'm going to show you another fancy command to get some demographics values and into this data set, which is called glimpse. Okay. I need to execute it here. Okay. So now if you see, we don't see 1, 2 in the gender field. We see instead male, female, and so on. And again, for the race and ethnicity, now we see the actual race or ethnicity instead of having them as number. Now, if you do this, it will make more sense if I say, give me a summary of, let's say, race and ethnicity. And when it summarizes the data, it makes more sense for me that, okay, I know I'm looking at 74 Mexican Americans and 18 other Hispanics, around 103 non-Hispanic Blacks, and so on, instead of just having them coded as numbers, okay? And the other thing is when you say glimpse, it also gives you a little more information here about the number of rows and columns in terms of dimensions, the number of variables you have, and the number of entries that you have in your data. It's very important to check the number of entries, especially because sometimes when you're working with really huge data sets, it takes a while to load up everything into the R environment, and sometimes it's not loaded. And before that, you start your analysis. So that you need to be careful on that. So that's why I'm stressing every time that check the dimension of the data that you are working with. Now, going forward, what is ggplot? Because we are now going to use ggplot for all our visualization going forward. Okay, so the GG in ggplot stands for grammar of graphics. And the basic idea is this: when you plot the data, you are creating a virtual representation of numeric or categorical information within a coordinate system. Okay, we have seen a couple of them. So let's say the most basic example is a scatter plot. The position of a point on the x-axis reflects one variable, and the position on the y-axis reflects another variable. Okay, that works well for simple examples, but often we have a large number of parameters that we would like to display. So ideally, we want a clear and a flexible framework that maps these arbitrary variables to arbitrary visual elements or aesthetics. Okay, and what are these? It could be, let's say, X position, Y position. It could be size. It could be color. It could be the shape. It could be transparency, and so on. Okay, so this would let us rapidly explore, like the ggplot will, with that we can explore different ways of looking at the data. Helpful. Okay, so ggplot provides this sort of a framework where you can have a clean mapping of the variables to the output. In other words, ggplot maps data to aesthetics, and the way it does it is, we'll see, it does so in layers. So there are several types of layers that I'll talk about. For example, you will have a geometric object, you will have statistical transformation, and you will have position arguments, and so on. Okay, so we can see how all this works by examining a syntax. So the way we initialize a ggplot is, so far we have been doing it with different commands like hist or bar plot or plot and so on. But here we always initialize a command with like this. Is it visible? Okay, ggplot. And then we go on adding layers with instructions for mapping. Like, what do we want to map? We want to map, let's say, the colors or the shapes. So we will keep on adding to this base command, which is ggplot. And we can also add other functions to customize the graphics. So it gives you a lot of flexibility. So the first thing, okay, let's do some plotting with continuous variables. Okay, so for example, if I want to use scatter plot, these are one of the most commonly used for graphics in these kind of data set where this is a laboratory medicine data set. So scatter plots are fairly common there. So if you want to create a scatter plot, let's say to look at the relationship between iron and transferrin saturation, like two of the variables here. And we want to use ggplot and some mapping onto the parameters, and also maybe a geometry with which we can make the plot pretty. We will, let's see how do we do that. Okay, so I'll give you a template for scatter plot using ggplot. Okay. So here's a template. We have ggplot, and then we'll pass the data. And then we'll use this plus sign to add on the layers as we want to customize the visualization. Okay. So, okay, let's fill it in. Okay. And run the code to see how exactly it works. All right. So what I'm going to do is I'm going to say ggplot. Okay. And this time I'm exactly going to use, I'm exactly going to use the same syntax that I have given here, but this is optional. Even if you don't say data equal to something, it is automatically, it will understand that it is the data set that you're passing. But just here, because I have given that template, I'm going to use the same thing. So I am saying to ggplot, this is my data, okay? And then, because we are going to do a scatter plot, I'm going to add this layer, which is called geome point, okay? And I'm going to add some aesthetics here, which is by a function called AES here. And what I'm going to say here, pass two arguments, what will be my X and Y. So let's say, just because we said we want to look at iron, and then okay, and we need to close the bracket, right? So let's see what it does here. Okay, I think. So one thing to be careful here is it is case sensitive. So just because I didn't pay attention here, so it didn't run and it gave me that error. So it's case sensitive. Okay, so notice that our ggplot command had three parts. The first thing is the data, the object that we are passing, and then the aesthetic mappings, X and Y, because it's a scatter plot in this case, and then a geome function. So geome has multiple functions, but geome point is for rendering the geometry. This actually doesn't look like our ordinary function calls, but you can think of the plus as a way of saying, okay, add this geometry rendering layer to the plot that I just made, okay? And the aesthetics that are specified within AES calls are where the real fun starts, okay? Why I say so, the aesthetics for X and Y can be specified in a global AES call in the main ggplot as well, okay? Or locally as within the function call for each layer. So right now it is within a layer, but we could also specify it globally when we are calling the data set, okay? So you have to be careful for the global versus local settings here. So there is some sort of an inheritance, mappings and data that appear in the global part, which is under ggplot, will apply to every layer that you keep adding. But mappings and data that appear in a layer or function, for example, here, will add to or override the global mapping for that layer only. So there's a little bit of distinction here. When you define it with the main function ggplot, it becomes setting. and it applies to everything that you keep adding, any layer that you keep adding going forward. But instead, if you don't define it here and you define it within one of the layers, then it becomes a local setting and it applies only for that layer. Okay. So for example, let's look at the same transparent saturation versus iron again and map the ethnicity. And we will now do a different setting here. We will specify, I'll copy this from here. Okay, and now I am going to move this as a global setting instead of a local setting. Okay, so I'm going to add it here to the main function. And then I'm going to say geome point. And I'm just, I could add some colors just to show you some variation here. I'm just saying that color it by the different ethnic groups that we have in the data set. So okay. Now, if I execute this, what's this here? Okay. So remember I was telling you that it has some default settings that it will pick up the colors. And for example, here, we didn't specify it to bring up a legend, but it did. Okay, so these are some of the nice things that ggplot offers you by default. Now you see, coming back to the point, if we now we have mapped the color in a different setting, and then we have called the AES here, right? So you can go ahead and customize this further. And just to mention here, like I think there are 6 different types of lines, and I think more than 20 choices of the symbols and shapes that you can use. Like some of them, some of those shapes we have used earlier, like I think we used 19, 8, and so on. So these are like different types of shapes. If I remember correctly, 16, 19, 20, they are all different types of circles. Some of them will be an open circle, some of them will be a filled circle, and so on. So let's figure out how the color and fill, these 2 parameters, they work together in different models. So starting with the default shape, for most of the geomes, the default is 19, which is a solid circle. Okay, the shape 19 only respects color, but whichever color is specified is used for both fill and the stroke. Stroke is the border. I'll show you what I'm saying here. Now it's the same plot, and I'm just saying don't do this coloring by the ethnicity, just color everything blue. I don't want to have it as a factor of different ethnic groups. The first basic setting. So just because I said color all my points as blue, and it did. Now, if I modify the same thing, let's say I want to have the same symbol, and I'm going to add here, let's say shape I'm just going to go by the default first, shape 19. Fill blue is okay, and okay, color blue, and I can say fill red. Okay. If I do this, it didn't change because for this particular shape, these two things doesn't work. But now if I, let's say, choose a different shape, you will be able to see the difference. So now I have a border of blue, and then the filled in part is red. So just to remember here, it doesn't work for that particular shape, which is 19, that's a default shape, but for any other shape, you can go ahead and customize the plot. Some of the other things that we could look at, like we can also combine these aesthetic mappings. Let's say we have seen it by the ethnicity. Now, if we want to layer it also by the gender. So basically what I am saying is one is the color, and now we have another variable, which is a shape. So I can make use of both of them. So I could, let's say, use color for the race and ethnicity, and I could use the shape to show me the gender on the same plot. Okay. So again, it's the same plot here. Okay. And then we are going to add a layer for geome point. And now what we are going to say is the aesthetic thing. What you have to do is color it. We are adding two different aesthetics here. Color it by the race or the ethnic group. And I also want to select different shapes for the two genders. Okay. And then I'm going to say, pass some arguments about the size and things. Okay. So I'm just going to bring this up again, just to show you what's happening here. So this was only, we have used the use aesthetics to color it by the race and ethnicity. And now we are adding one more argument here to color it, also distinguish between male and female gender here. So it should, okay, it's a bit slow. So now you see here, so if I look at this, I can say that, okay, this individual is a male and he belongs to Mexican American ethnicity. And so it's like I am adding more information to my graph by using different options and layerings. Okay, now let's talk a bit more about colors. So if you look at the R palette, there are, I think, more than 600 colors available. And you can specify colors in many different ways, just by the name, by the hexadecimal code. And there are 50 different color palettes available. Like when you are doing, let's say, a heat map or a gradient, you want to know which color would look as the best contrast. So those palettes are also available. But just a quick tip here. Instead of, though I have used here the colors by the names, but I would suggest that always go with the hexadecimal code for the colors, just because they are not going to change if any other settings changes. So those are the best ways to specify the colors. And I'm going to give you a link here, which will have where you can access a full sheet of colors. And okay, so if you go to this website, you will have plenty of choices to choose from. And there's also a very uh, great package that we can install, which is called our color viewer, that has many color palettes that are useful for visualizing ranges and figures, especially in heat maps. Okay, I'm going to show that. And also, let me just, I think I have that site here. This is another reference that you could use. And then if I now load this library, color viewer, just as a way of mentioning, I am loading it just because I have installed it at some point. But if you just load this library, it might not appear. It might not be there in your installed packages. So if it doesn't, then all that you have to do is go here and install, look for the package. Okay. Since because I have used it earlier, so it's already there, I could that package to use it. Okay, so I have loaded it. Just to give you what we have inside this is, okay, it's good. It's just
Hello, excuse me, ma'am. Hello. Okay. For the participants, we are having some technical glitch, so I request everybody to please show patience for 2 minutes or so. Just give us 2 minutes to get prepared again. Thank you. Ma'am, can you hear us now?
I can hear.
Okay, so you were not audible to us, for the past 5 minutes, I guess.
Okay. Can you tell me, like, uh, till where did you, till what point I was audible?
Ma'am, you were audible till.
Okay. Okay. Sure. Are we back online? Okay. Sorry about the technical glitch. So all that I was saying here is that I was able to load my library R color viewer just because I have used it before. If you haven't, it's the first time that you're using, all that you have to do is go to packages, under install, go and look for the package, and then click install before you could load it. Okay, and then this command brewer.pal.info will give you a list of different palettes that is present in this package, our color viewer. And from here, you could actually choose what you need. Sometimes it's a general specification that you need something which is compatible with colorblind people. So you could choose a palette accordingly. Depending on how many colors that you need, you can go ahead and choose. So there are many options for you to go ahead and choose from this palette. So let's try this out by looking at our data set again, and let's look at the total iron binding capacity by the race or ethnicity and age. Okay. Okay. So I'm going to minimize, make it little. Okay. So we are using the same plot, same data set, and I will get the command first, and then let's go through this. Okay. So it's the same syntax that we had before. It's the same template, ggplot, passing it the data set, and then we are setting some of the aesthetics in a global setting, that we are saying our X will be age in months and Y will be the race and ethnicity, and fill it by this variable. So this is our global setting, okay? And then we are layering it. We are saying, the color that we should have is blue. It's you can play around. It's a continuous scale, and just because I want to show you the use of this color viewer, so I'm saying, I need 5 different shades of green. So this palette will help me choose 5 different shades of green. And I could set a different theme, but I'm just saying here, keep it minimal. Show me. So this is the grid panel setting. This will be the title, and I will just not use this for now. Okay, let's do this part. See what do we get. Okay, so this gives me a customized plot like this, where I have age in months and the y-axis label here, the ethnicity. I could go ahead and customize. I could say, no, I don't want a shade of green. Maybe I need a shade of orange. Okay, so it's not available. Really? What's going on? I think there's something wrong, but it shouldn't be the case. Let's stick it with green. But you could definitely change those options. The other thing, let's say if I don't need the labels here, I could just say Y label and just put it as blank. So then the Y label will disappear. And if similarly for the X labels, and remember, I'm just adding them as layers as I'm going to my base plot. No, there is something wrong here. I think it's not loaded, maybe. Oh, what's the problem? Oh, yeah, now it should work. Oh, I know. Probably I have to say here reds, because it's going to, that's how it is mentioned in the package. Should bring up, yeah. So this is something which is specific to the palette here. So just because it is specified as red, so I need to I had to add this extra S. And then I have added the title to this plot. Now to add the title, just because it's a GG plot, so I have to use a function called GG title. Okay. And then let's add some data manipulation to customize what we are illustrating. So let's say if we want to look at multiple variables at one in a time series type of plot, okay? So just to have that visualization, probably you need to do a bit of data manipulation first. And then once the data is ready, then you can plug it into a ggplot and layer it out the way you want to visualize, okay? So I'll give you a quick code here, which is easy to follow. Okay, so what I'm doing here is I'm taking this data set and I am piping it and then I'm filtering this data set, and I want to select only the males in the data set whose ethnicity is Mexican American. When I filter this, then I want to pipe it further. I want to group it by the variable age, and then I want to mutate. I want to calculate mean for each of these measurements that I have, and I want to label these the output as mean underscore whatever is the variable name. And once I do that, I further want to pipe it, and I want to select age in months and the variables, and then further pipe it into a slice command, and then ungroup it. Okay. So you could actually do this depending on what do you need to look at in your project, but like some of the things that you could do, right? So now we haven't plotted. It has just, if you look here, it has created a data matrix for us to look at. We could So we have basically prepared our data that we want to plot. Okay. Now we could. So if you want to, so I'm going to show you how we are going to use the tidyverse package and pipe it. But the other way would be to use another function which is available, which is called reshape. Maybe some of you are familiar, which basically is going to reshape the same data in a way that we could plot it easily. So I'm just saying MMA long because what it's going to do now when I execute pipe this, it's going to give me the data one measurement per row. So let's look at this. Now, instead of 20 observations over 4 variables, now we are looking at 60 observations. So if you want to look at this long data that we have generated. Okay, so basically we have, we can open this. So it will open it up here. So what we can see is that these are our markers, these are the values that we are looking at, and this is their age group. Okay, and it has done it for the entire data set from instead of having them in a condensed format here. Now with this, let's plot the graph from this subsetted and reformatted data. Okay. So I'm going to close this here. I'll go back to the script. Okay. So I'm using ggplot again. Now my data set, which I want to pass, is this MMA long format. Again, setting the global variables here, and I'm going to use these markers as a way to group my data. And now I'm going to plot a line. And these are my colors. And I don't want a background theme, so I've kept it blank. I've not passed any argument here. So basically, it has plotted the results over the age for these 3 different markers, and it has computed the mean for each age group and plotted them. Okay. So this is basically another type of continuous variable that which you could have, which you can visualize. And I mean, these are just a handful of visualizations that I'm trying to show you, but you can do many more. And you could play around with different types of visualization. So next, I'm going to show you how do you plot the discrete variables. Okay. So let's say if you want to compare counts or values across discrete variables, we need a different set of plots. Okay. So, and we can also do this using ggplot. And all we have to do is we have to specify a different type of geome instead of the ones that we have been using, like geome points or geome lines. So let's talk about the bar chart. So there are 2 types of bar charts. One you can specify by saying geome underscore bar, and another one is geome underscore column, like abbreviated as col. Okay. So what geome bar does, it makes the height of the bar proportional to the number of cases in each group. And if you want the heights of the bars to represent the values in the summarized data, then we actually use the other one, which is geom_call. So let's say geom bar uses something called stat count. By default, it counts the number of cases at each X position. Okay, whereas geom call uses a stat identity. It leaves the data as is. Okay, this means we do not need to provide a Y variable for the geom bar, but we do have to provide that for when we use this function geom call. So let's take a look. Since the data that we are using is not summarized into counts, so we will use geom bar. So if we have the bar fill by categorical variable, we see a stacked bar plot showing the relative numbers from each group. The categories are plotted based on the order of the levels. For example, we could choose any of the ethnic groups. Let's see, how do we use this? Okay. So it's, again, the same format, right? ggplot, the data here, some global settings. And now we are saying that we want to group, the X will be the race or the ethnicity. And we are going to have these bars, and we will fill it by the gender. And these are other adjustments. And again, this is basically to hide the X-axis label. Let's see what happens if we bring up this. Okay. So here, note that we used fill here to color the bars rather than using the command color that we have been using earlier. Okay. So let's see what happens if you use color instead of fill. So I'm going to just copy this again and replace fill with color, because they have different usage. So that's what I want you to get here. Okay. Again, it's by the gender, but instead of fill, now we have replaced it with color. See the difference? Now it has only colored the borders. It hasn't filled in the color. So that's the difference. If I use fill, it will fill in the bars as shown here. But instead, if we just use color, then it is only going to color the borders. So that's the difference in usage between fill and color. Okay, now if we don't want a stacked bar plot, okay, which is the default, we can specify an argument of position to change this arrangement. One of the cheat sheets that I have given you in the reference will show the effect of the different positional arguments. Okay. So there are commands like dodge and fill. They are commonly used for bar plots. Okay, let's create a bar plot where these bars from each category are not stacked, but rather placed next to each other. Okay. So note the difference here. What I have done, it's exactly the same thing, but what I have said, I have added one more argument saying position equal to specify it as dodge. So now this is going to do the trick for us. Now these bar plots are no longer stacked, they are just next to each other, okay? I'm not getting into these because they are really simple to follow, but just as an example, it will just change the angle of the text, something you guys can explore yourself. And then you could also adjust the height to place it the way you want. So I'm going to change it back here. So this is another thing which you could go ahead and play around with. Okay, now let's say for some visualization expert would suggest that, the orientation of this bar plot is not ideal. Given the long names of these race and ethnicity, we shouldn't view it this way. In this case, we can do a quick transformation of the axis using another argument called quad_flip. Okay, so it's going to be still the same thing. I'm going to copy this. Okay. And I could still have this one here because I don't want any labels. And I am going to add one more command, which is this. Okay. And what this will do is now it's going to change the orientation of this graph, this plot. Okay. So this is another type of customization. And of course, if we want to go back to good old histograms and get their own geomes, so for that, it's a different geome type, just because it is ggplot. So it'll have something like this, and we will make use of this if you want to draw a histogram. And again, we can fill by categorical variable, currency and position arguments to visualize the overlap in the distribution. Okay, so let's see if now it's a ggplot way of doing a histogram. Okay. Okay, let me just bring this up. Okay. So just note one thing, the ggplot command remains the same. What changes is the layers that we are adding. So it's the same data set. Now we are adding a different layer, which is geom_histogram, to have a plot where it will show us a histogram. Okay. Now this is a histogram where I'm looking at the counts of this variable, and I have factored it by the gender. I could do the same thing. If I want to look at, let's say, the race. Okay. It's suggesting me to use a different bin, which I could if I want to, depending on the data distribution. Okay. Now, let's say, now we have looked at the histogram. Now, let's say, no, I don't want to look at the histogram. I want to change this histogram to a density plot. And I want to customize it. I don't like my lesion here, and I want to move it to a different position. Okay, so how do we do that? Let's say change the histogram to density plot. And also we want to shuffle the legend position. And oftentimes we actually need to do this just because we want to fit in the figure or the subfigure into the space that we have for our publications. We don't have, we are basically wasting so much white space here, but instead maybe we want to put them at the bottom. Okay, so there are two things we are doing. So note here, now instead of geome histogram, we are using another function here, which is geome density, to add this layer. And the other thing that we are specifying, because that goes under theme, the legend position, and we are saying place it at the bottom. We could have it on the top or wherever you want. There are several positions where you could place it. So now it transforms this histogram into a density plot, and you have all your lesions at the bottom. Okay. So basically, the message here is layer it on. Okay. So ggplot objects can be layered and layered upon, including multiple geomes, labels, custom scales, statistical results, and many more things. So we have seen a couple of examples, and now what I'm going to, let's add some lines, okay, to the histogram. Let's say the one which we have done before. Here, oops, here, okay. So we want to add some vertical lines just to show some of the results, probably maybe high or low or whatever. How do we do that? Okay. Let me write it here. Okay. Let's add lines to our histogram plot. Maybe showing the reference range limits, because these are lab data. Okay, and label them high, low. So, okay. So again, it's the same template, ggplot. I'm asking it to do a histogram, exactly what we did before, factor it by gender, and then I'm now adding some lines. My x-intercept is 50, and I'm saying this is a line type, which is a dotted line. What is the size? And then I want to add it. If I just do this, it will draw the line. And then I want to annotate, and I want to add some text here. I'm specifying the coordinates x and y where my text should appear on this plot, and what it should, the text, what text level it should have. It should have text level low and the angle at which the text will be oriented. So I can basically control every single thing on this plot, including what I want to put, the size of the text, the angle of the text, and whatever I want to, however I want to customize. Similarly, I want to add another line, vertical line at intercept 120, and I want to say that if it is at beyond that range, it is high. And also I want to customize the lesion position. I don't want to waste the space here. I have a lot of space inside my graph. Maybe I want to bring it inside. And that's what these coordinates are going to do for me. Okay. Let's run it. See, so now it's saying that if the iron level is below this, it should mark it as low. Anything beyond this is high. And then it has also factored it by the gender. So for each gender, maybe the ranges, whatever you want to customize. Okay, so those things are you can have a good visual representation of that that kind of data here. And other options for adding labels to plot include, you could also play around with functions like geome label. That's something to explore. And then there is also another geome function, which is geome text. Okay. Now, let's say, slightly getting into the stat side, let's say we can add a best fit regression line to my scatter plot. I'll not get into the statistics part, but I'll show you how do we do it. There is a so what we are going to do here is that add a best fit regression line to a scatter plot, just because we are assuming it's a linear relationship. So we are going to use this linear model here. And so I basically want to introduce you to another geome rendering, which is geome smooth. And if we run this, we will have, you may have to optimize it a little further, adding some options for your data, but this is a very high level basic plot for fitting a regression line to your plot. Okay. Now, let's say to add the best fit line, and we also want to have the R-square value is a bit more complicated, but there is a way to do it. Let's say, it's not my function. I have just adapted it from one of the websites. I'm going to give the reference here. So I've adopted the function for computing ggplot regression, which I'm going to paste next. Okay. Okay, so this is a function. Maybe there are more details if you go here, like what's the logic behind this regression function, but I'm just going to use it for demonstration. This is a function, and then if I want to use this function and fit a linear regression model to the data set that we are working with, so see here under environment, now I have this function added. Okay, so any function you add, it will appear here, even if you write your own function. Should be doing it here. So what I'm doing is I'm calling this ggplot regression function. I'm doing a lmfit, which is a linear model, and passing my variables from this data set. Okay, if I run it, here. So it has basically given me some stats here. What is my adjusted R-square? What is the intercept? And what is the slope? Okay. Moving on, so layering plots and stats output, and then you could also layer grouped box plots, okay? And also have some statistical summary, for example, mean values or median values. And this can be done using some of the stats function. The argument is stat, and we have to pass this argument within geom points. I'll show you how do we do that, and then we can actually call a stat summary to get that. So I'll just say it's another example here. Okay. So again, it's a ggplot, setting up the global settings, and we are going to do a box plot and a geom point. So now we are using two different renderings on the same visualization. So all that we are saying is do a box plot and also show the mean using this geom point. So it will compute the mean using this function here. So I'm using the stat function to summarize. And what kind of summary I want? I want mean. So I am specifying using this function .y because I want mean for my y-axis. And then select color it red, and this should be the size. And I'm using coordinate quad flip just to have it in a different orientation. So if you see, so basically there are two different plots which I have added as layers. So the first one is the geom box plot, and then the next one is I have added this mean function. And okay, we can do it again just to show you that what happens if we just use one of them. I'm going to take out this part. So it's just going to give you the simple box plot without showing the mean. So now it's not showing the mean as it was showing just because we have added one more rendering using geom point, and then we have added some stats that show me the mean. And by default, the box plot, it shows the median here. Okay, and now to have multiple layering, we could even go beyond this, like not just the we can customize it further. Okay, this is another example. Okay. Should just say layering multiple plots. Okay, so it's a different type of plot that I'm going to render. So, okay, ggplot, dataset, global settings, then default do a box plot. And once you do the box plot, do a dot plot. And what that dot plot should have, I'm specifying the bin axis should be the y variable from here, and giving them the color, passing the arguments for color, dot size, and so on. I don't want the legend here, so I said like I don't specify the legend position. I say none, because by default ggplot will bring up a legend. And then some titles and subtitles, and then whatever, and then again a quad use of this quad flip function. Okay. So basically, it is a box plot plus a dot plot. Okay. Now, creating and combining multiple small plots, that's also another feature of ggplot. So it has several faceting function to divide plots into subplots based on the categorical variable values. For example, if you're doing a PCA and you want to look at the scree plots, that's one option of faceting the plots the way you want. But actually, there is another package which provides this function, which is called grid extra. I should write it up here. Grid. This is the package which is needed to arrange multiple independent plots into a single figure. Okay, so instead of, let's say, instead of overlapping the density plots by race or ethnicity, now we can create individual plot for each group using this faceting function, which is facet underscore grid, which will arrange the subplots into columns or rows depending on what you have specified. So the data and the plot type may dictate the orientation, which orientation is the best. So let's see. So let's say we want to view them as columns first. Okay. So what we have here is, again, ggplot data, and then we are saying it's a density function. You could also pass on other arguments if you want to, but what I basically want to show you is how you facet it into grids. And I'm saying that group it by, so faceted by this race or ethnicity, and wrap it, and then whatever is, however I want to space them. Okay, so if I run this, so now it is instead of an overlapping density plot, which we have seen earlier, now it's individual density plots, but they are all presented as subplots to a big plot, to the main plot. Okay. Now the same thing we could, if we want to do it in a different orientation, let's say as rows, that's also possible. The only thing we need to change is oh, sorry. I think it has multiple glitches today. Okay, that's good. So the difference is here. Okay, I have said it. I have placed the tilde here, but now I'm saying tilde and a dot later. Okay, so it's now, it will be the same plots but in a different orientation. Now, once you have generated so many plots, I'm sure you would like to save it, which is a very important part of generating this visualization. So again, if you want to do it by the graphical user interface, you could just click on this export function here, and you could save it as an image, save it as a PDF, or you could even copy it to the clipboard. what I'm going to show you here is oftentimes there is a requirement of the journal about the resolution and the height and the width. You could also specify uh, the values here depending on what you need. And then you could say save. You could choose the path or the directory where you want to save your plot. And you also have options like there are several different options here: JPEG, TIFF, BMP, PNG, whichever is suitable for your analysis. You could choose that. And that's the easiest way to save. I'm not going to save it here. That's one way to save plots. The other way is that you could use a command to save your plot using ggsave. That's the command. And you could basically pass on the plot that you want to save to this and specifying the format, and it will save it for you. Another thing which is very interesting here is when we are talking of layering, I could do this. I could, let's say, assign my first plot. Okay, I'm just going to move this. If I run only this part of the command, so now it has saved this into a variable called p. Okay, but it hasn't really done the faceting yet. What I could do is I could say plus and then add on the other layer here. And if I do this, I should get back the same plot. So sometimes we want to reuse, we want to reuse a part of the visualization for several subsections. So we could save it in a variable and then add the extra, the other layers by as we need it. So for example, we could, here we have done it by the race and ethnicity. We could also do it by gender. So I don't need to redo the first part. I could just store it in a variable and keep reusing that part. Okay. And after all this, let's say we have done our coding. And the thing is, we need to clear the environment, because let's say you want to do the next thing. We have so many things loaded in the environment here. How do we clean everything that we have loaded? Okay. So the command is rm, and I can say here list is equal to ls, right? So everything that is listed here, remove everything. Okay, so it is basically a command to say clear environment. Okay, and remember at the beginning, so it has just cleared the environment, but it still has certain things here. And what are those? The packages that we have loaded. Okay, and now it's going to be super easy just because we have used package manager. So to clear the packages, I'm just going to ask, remember if we go up, we have used this command here, pacman pload, to load all these packages, and along the way we have loaded many more. So I could just go down and say p unload, and then I just say all. So it's going to basically remove all add-ons. Okay. So everything that we have loaded, either using the package manager or we have manually loaded some of the packages, it has cleared everything. And the very last thing that you should know is how to clear the console. Okay, before that, there's one more thing. So package manager has unloaded everything. Let's say if you are not using package manager, then how do you unload or detach a package? So for that, the command is, you just say detach, and I think we only have the base package here, and the data sets in the base package. I could just use that. And I say unload equal to true. Okay, so it's going to unload the data sets. So detach is essentially the other way to unload the packages. It should go a little down here. Here. And finally, how will you clear the console? Okay. All that you have here, how do you clear it? I'm going to write the command, but I'm going to tell you the shortcut here. Okay, so this is the command you could type, but the shortcut is Control plus L. So if I do Control L here, it's gonna clean my console. Either I do Control L or I have to execute this command. So both will do the same things. Yeah, and I think, yeah, that brings me to the end of my today's talk. I'm happy to take any questions that you may have.
Thank you very much, ma'am. That was a really nice presentation. So, as for the questions, we can share the YouTube chat link with you in the Google Meet, so you can directly look at the questions. So, just a minute, ma'am.
So,
Yes, it would open the chat in the YouTube.
oh, I see. Okay,
Yeah.
it's coming up.
Yeah. So,
Okay, so the first question is, which one is better, R and SPSS? Well, okay, that's a good question. There are the way you look at it, right? First of all, R is open source, it's free. You don't have to pay for any licensing. And whereas SPSS, it has a lot of functionalities, but still it's kind of limited. And the thing, like the packages like ggplot2 and tidyverse, which really makes R so much flexible, and you can do anything that you would like to do. But frankly speaking, I do use SPSS for some of my clients just because they are more familiar with it. Especially they are from the medical field and they don't have much idea to go about programming. But it has limited functionalities, plus you pay for it. And whatever SPSS can do for you, R can do it for you, being open source. So, and you are not dependent. And SPSS, if you has perhaps certain limitations in terms of how much data it can load. But that's not something which you will face in R, as long as you keep checking that you have loaded the entire data set. I keep saying this because that's a practical problem you will face when you actually start analyzing big data sets. RStudio is the best in GUI. Yeah, so this is an environment. RStudio is the environment specially created for R, and if you want to use a graphical user interface, RStudio is the way to go. Can we do PCA? And oh yes, you have several options for doing PCA and cluster analysis through our software. It's just that you have to load the right package. And for cluster analysis, also you have various options like hierarchical clustering or any other clustering that you want to use. Yeah, there are several options. I didn't have the time to go over PCA. That's one of my favorite topics, though. Yeah, maybe some other time. But you can definitely do it through R. So, okay, so the machine learning, yes, you can definitely do that with R. It's not a limitation. But I would say that if you have to get into the area of machine learning, then Python is the way to go. And it has several packages, which is really friendly for machine learning. So, I mean, that would be my recommendation. So basically using that's what I said at the beginning, use the right tool for the right problem. So bring out that tool from your data science toolbox for the problem that you're addressing. So if it is a data mining and data visualization thing, R is more preferable in biological research. But if you are trying to do more of machine learning applications, you should use Python. Do we have more questions?
Yes, ma'am. You can just scroll through the chat and you can find more questions in the upper section.
Oh, okay. Oh, I started from the bottom, seems. Yeah, something like standard deviation in R, yes. You can, that's a part of the base R functionality, and you could do standard deviation just by I think it's the function is std, and pass the data to that function, and you should have the standard deviation to your data. Then let's see what else we have. Type packages could not find the function. That's kind of odd, but how exactly did you do? Did you go for the tidyverse packages? Did you go via the GUI route, or you use the command line version? But either ways, it should work. Yeah, I'm going to share the R commands that we did today. How can I write different types of commands in R? So RStudio is just the environment. So everything that you do is still R. RStudio is just providing you an environment where you have everything at the same place. Like you can see the variables that are loaded. You can have the help function, and also the script that you're writing the output of the script as well as the plots. So that's an environment which is provided via RStudio, but it's still running R as the programming language. I mean, there is only one way to write different type of commands. The more you practice, the more you get used to it. You can save the table in CSV format using MS Excel. Yes, you could save an Excel sheet as a CSV format just by choosing the option Save As. But I didn't go into it, but R has the functionality to read the Excel sheets as well. So when you read Excel and you have multiple sheets, you could specify which the sheet from which you want to read in the data. So that functionality is also available in R. Not necessarily you have to convert your Excel sheet into CSVs. But CSV is more like a text formatted form of the Excel. When you say, how do we introduce our experimental value in R to plot? So it's the same way that you have to have, let's say, a table where you have your experimental results, which you will read into R. Whatever format you save, is it a .txt, .tab delimited, or CSV, or even Excel? And then depending on how you want to visualize your graphs, you are going to use some of the commands that we have seen today, or maybe some more, and you could see the plots. Can you help me, Neha, if I have missed any of the questions?
Yeah, sure, ma'am. So we have a few questions from Yogita Lugani. She wants to know that can we use our software for bioinformatics figure like protein structure to get good resolution figures?
So if you're saying that you already have a figure and you want to improve the resolution, I mean, the easiest way, if you could use one of those Adobe Photoshop tools, like it helps you to increase the resolution. There is no point. So R is basically a statistical software, in a sense, like you can have the data and you can plot it, but if you, let's say, have a protein folding structure and you only want to use the resolution, I mean, this is not the best way to do it.
Okay, thank you, ma'am. So one more question we have is that Krupal Joshi wants to know that how to create a heat map with number of data sets.
Number of data sets. So we just did some heat map today. So just, okay, I'm not sure what type of data set, in what format you have it exactly with you, but the best way would be, all you have to do in a very simple one-liner way is use the function heat map, pass your matrix into heat map. Okay, so, but now you have to prepare your matrix. It cannot be a data frame, just because heat map, if you think about it, it's giving you the gradient of numbers. So you have to choose the variables that you are using for plotting this heat map. So for example, if I choose something like a gender, then there's nothing to represent in a heat map format. It's sort of a binary 1 or 2 or 1 or 0, that kind of a thing. So as long as you choose the right data format, you should be able to plug it into heat map and have the visualization.
Ma'am, she's talking about microarray datasets.
Oh, okay. So yeah, for microarray datasets, there are packages. I don't have it top of my head. I have done it sometime in the past, but send me an email. I can send you the packages which are specific to view the microarray data. There's something specific for NimbleGen custom arrays and stuff like that. I don't have the package top of my head right now.
Yes, ma'am. We'll share your email ID with the participants if you say so. Then, ma'am, we have one question that how can we save charts and tables for publication purposes, like in a high resolution? I guess I have asked this question already, but okay, yeah. So.
Sorry if I have missed that. So you're going to use that command ggsave. There you can specify not only the height and width, but also the resolution. So you can choose depending on 300 dpi or 100 dpi, whatever resolution you want. You can actually specify in that argument where you are using ggsave.
Yeah, so ma'am, that's all for the question side, I guess. Yes, ma'am. So thank you very much, ma'am. Ma'am, now Yasha ma'am would like to say something.
Thank you, Dr. Tannistha. That was truly a hands-on session, starting right from the very basic step of installation of R to reaching this far in data visualization. And I'm sure participants would be able to appreciate the power of R, and they would be able to apply this language in whichever field they're working in. Thank you so much.
Thank you.
So thank you, Tannistha ma'am. Thank you, Yasha ma'am. So with this, we complete our lecture for today, the first lecture for today, and we'll meet you again at 2 p.m. IST for our next lecture. That would be in continuation with the lecture we had yesterday of Dr. Amit Agarwal. So for people who has just joined us, can go to the link and just go through the last videos the sir shared, and we will meet you at 2 p.m. Thank you, everyone.
With the link, with the
Yes.
files already downloaded on the systems.
Also, the participants can go to the lecture and see the links and download the data sets which sir has shared for the hands-on session today, and you can go and just check it there in the description box. So thank you very much. See you at